Good morning, good afternoon, good evening, depending on where you're at. Welcome everyone to this exciting presentation. I'm Matthew Hestan. I manage the PacBio's field bioinformatics teams in North and South America. Thanks for joining us. I'm joined here today by my colleague Yinan Wan, who's Senior Product Manager in our Applications and Partner Software Group, and also Billy Rao, who's Senior Staff Scientist focused on bioinformatics. A couple quick housekeeping things. Please note that both of today's presentations have been pre-recorded, but the talks will be followed by a live question and answer session with the speakers. You're also welcome to submit your questions during the talks by typing them in the area provided on the attendee control panel, and we'll get back to them during the live Q&A. We've also uploaded a complimentary piece of literature to your control panel, so please feel free to download it and take it with you. So with that, let's get started with the first talk from Yinan on an introduction to the pipeline and the underlying tools.
My name is Yinan Wan. I'm going to introduce a set of latest developed tools and workflows from PacBio and how this is going to improve the experience working with HiFi data. HiFi reads have been known for its advantage of being long and accurate. Beyond that, we also get excellent even coverage. There's little GC bias in our sequencing data. It allows us to sequence all throughout the genome. This completeness also includes methylation information, which you automatically get from HiFi whole genome runs for free. Like mentioned earlier, HiFi reads have been extraordinarily accurate. The long reads length also help with allele resolution phasing. This gives you a full picture of the genomics, and you can explore all the variants that are occurring in your sample. While most people may be familiar with SNVs and small indel callings with short-read sequencing, the HiFi genome is a truly representation of whole genome, as the long, accurate reads from HiFi excel at not only small variants, but a list of complex variants and events, including structural variation, methylation callings with no additional library prep or sequencing, segmental duplication, phasing of the 6 megabase deploy human genome, and overcomes the issue of missing hundreds of millions of bases and over 400 medical relevant genes represented in the dark region, which are inaccessible with short reads. It really shows that not all genome sequence data is created equal. Our journey explore human whole genome only begins with the high quality HiFi reads getting off the machine. The PacBio research team is constantly innovating with new tools that uncover more from HiFi sequencing. Today, if you go to the PacBio organization on the GitHub, you will find a good list of tools serving applications at all areas, such as human genome analysis. Even if you are new to HiFi data or another specific application area, we recommend you to go to our computational tool webpage and grab a demo data and jumpstart exploring the technology. Here's an overview of what you can do with HiFi human whole genome data. The journey usually starts with alignment, where we provide aligner pb-CpG-2. pb-CpG-2 is Minimap2 with a wrapper aiming at optimized performance to best support PacBio data with recommended parameter and sorted output. Deep variant is a well-known small variant caller developed by Google, including a PacBio model, which has been trained with HiFi data showing excellent performance and validated by our research team as well. Moving on to structural variants, that is large insertion, deletion, duplication, inversions, and translocations. We have PBSV as the SV caller. For small variant phasing, WhatsApp has been a well-accepted tool from the community. All the tools goes up to here. You will be able to access it through our fully supported SmartLink software suite in HiFi mapping or variant calling analysis workflows. For more advanced variant type, we also introduced target for tandem repeats and paraphase for segment duplications. For deletions and duplications larger than 100 KB, we recently published HiFi CMV as the depth-based caller for those events. We have Jasmine for methylation calling with added tag to the band files. pb-CpG-tools take one step further to perform pile-up and methylation probability based on those evidence. For those more advanced tools, as well as those already in SmartLink, you will also be able to access through PacBio GitHub, like we mentioned in the slide earlier. For the rest of time, I would like to dive into some of our more advanced and recently developed tools. Let's start with Target. Tandem repeats are genetic sequences that are repeated in series. They can also grow in length from parent to child. More importantly, tandem repeats have been linked to many neurological disorders such as ALS and Huntington's disease, in addition to the #1 cause of inherited mental intellectual disability, Fragile X syndrome. Short-read whole genome sequencing only offers limited repeat expansion resolution due to the read length. To fully utilize long reads to interrogate tandem repeats, we have developed a new software suite that is designed to provide best-in-class repeat expansion solution, named as Tandem Repeat Genotyper, or Target. This tool will enable targeted analyses of short tandem repeats or variable length tandem repeats, as well as the visualization of the results. We have an overview of this tool in the next slide, but I also want to mention the paper categorized this software was recently published in Nature Technology if you are interested in additional information. At a high level, the caller takes aligned HiFi reads as well as a bat file containing location information of the repeat expansion in question, as well as the expected repeats from the reference genome. The tool output a VCF containing information including the consensus sequence and the accurate repeat counts at a location, methylation level at the site, motif construction, and related interrupted sequences. All of those are reported at allele level for each genotype, and is all critical information to downstream to fully explore the mechanisms and impacts of tandem repeat mutation on disease phenotype. Also, to help user better understand the result, the tool also comes with a visualization tool, TRVZ, to display events at location per genotype, including the repeat structure, interrupted sequence, and supported HiFi reads. Moving on to Paraphase, which is a caller specialized in segmental duplication region. Segmental duplications are duplicated blocks of genomic DNA ranging in size up to 200 kb. They are known to be informatically challenging as alignment and variant calling are limited due to high sequence homology with paralog or pseudogenes, but also comprise 7% of human genome, and many clinical relevant genes fall into those regions, which we often refer as dark region, and are often hotspots for structural variation, including deletion, duplication, and conversion. This is why we developed Paraphase, a tool to provide a way to solve genes in segmental duplications with the advantage of HiFi reads. Paraphase works by first extracting reads aligned to relevant regions and realigned to the gene of interest, then phase them into haplotype. By doing so, it solves the problem of alignment difficulty due to sequence homology, and allow us to examine all copies of the genes in gene family, as well as cause CMVs and other variants, like we mentioned here. A JSON file will be provided to summarize the haplotypes and variant codes for each of gene family in each sample, and users will be able to infer the disease and carrier status from the information there. So to further illustrate the performance of Paraphase, here is an example of SMN1 and SMN2, which are genes associated with spinal muscular atrophy. Lack of functional SMN1 gene will lead to SMA, and the copy number of paralog SMN2 determines the disease severity. Also, both genes are 30 kb long genes and about 99.9% similar. So it is a challenge to understand the situation of the two genes in a sample. Short read sequencing also results in more than 80% of the reads to be MapQ of 0. Now with HiFi data and paraphase, we're able to determine the number of copies for each genes and the paraphased variant at each haplotype. Like what we're showing here in the IgV report, users can take information about the haplotype for each genes and further inform the disease or carrier status. For larger deletions and duplications, we also introduce HiFi CMV. For structural variants, we already have PBSV in-house as a break and caller. But some copy number events, especially those larger than 100 kb, may not have a breakpoint signature, and we found it is better to be identified using read depth. HiFi CMV works by taking a line band file to estimate haploid depths per chromosome set, and further detect copy number signals by comparing read depths. During the evaluation of a set of clinical relevant CMVs, the combined usage of breakpoint and read depth signal identifications are able to retrieve all the events from the set. A VCF containing copy number variant calls will be provided as the primary output, as well as a bigwig and bedgraph files for visualization purpose. Next, I want to introduce a new phasing tool, HiPhase. Historically, we have seen third-party tools like WhatsApp to have decent performance in small variant phasing and well accepted by the community. While this is encouraging, we're still seeing gaps in those we want to further address. This includes joint phasing of all advanced type of variants besides SNV and indels, starting with structural variants, as well as there's breaks in phase blocks caused by alignment issues. With HiPhase, we're now able to address those gaps we just mentioned, as well as increased performance and accuracy and a few other quality of life additions. HiPhase takes in alignment files from PBMN2 and produced a phased VCF following VCF spec 4.2 convention and a haplotagged BAM following the same convention as WhatsApp, so to allow users to upgrade to this new tool and enjoy all the improvements with minimal integration effort. The last tool I want to mention here is pb-CpG-tools for methylation statistics. As now we all know, HiFi data comes with methylation information without the need of separate library prep. The 5MC color marked the methylation status by adding standard BAM tag to the BAM from the instrument. Here, the pb-CpG-tools further performs pile-up to generate methylation probabilities for each site and haplotype. The tool output a bat file to capture the methylation score for each site, and accompanied with a bigwig file so users can pull in both the BAM and bigwig file to visualize the methylation information in the IgV. With all the advanced tools we recently developed in-house to better understand the human WGS data, the next reasonable thing to do will be to have a pipeline to call them all at once, so to provide a comprehensive view of the data. Here we present the PacBio WGS variant pipeline. It is a pipeline connect all the major tools applying to human whole genome analyses, starting from alignment with all the major variant callers we just mentioned, to some tools to help people get started with annotation. The pipeline comes with a set of pre-configurations for all major clouds as well as on-prem HPC, so to lower the effort for people to start their first analyses. It is implemented in the workflow description language and primarily distributed on GitHub as a command line tool. But you will also be able to access through our PacBio compatible partners with a point-and-click solution. That is to say, If there's a limitation for users to establish command line tool or looking for compute infrastructure for solid performance on data analysis, we have 4 partners offering analysis platform on the cloud with ready to run WGS variant pipeline for users to jumpstart understanding their data. Later last year, we also welcomed 2 tertiary analysis partners to provide services interpreting the human whole genome data, especially in rare and inherited disease research areas. Now, with all the advanced tools, pipeline, and effort from our partners, we have all the pieces together to be able to fully explore the human genome. Majority of the information and user guide from each tool and pipeline can be found in the corresponding GitHub repo. We also published the application note to fully describe the details on whole WGS variant pipeline. If you are interested in additional information about our partners, you can also visit the PacBio compatible partner webpage.
Thank you for having me. Hi, my name is Billy Rowell, and I'm a staff scientist at PacBio. Today, I'm going to tell you a little more about the PacBio WGS variant pipeline, including how to set it up, how to run it, what outputs to expect, and what improvements are planned for the next year. As you heard from Yinan, the PacBio WGS variant pipeline is primarily distributed on GitHub at the address on this slide, but you can also launch it from DocStore and on the platforms of multiple cloud vendors. This workflow description language, or WDL, pronounced WDL, pipeline can run in the cloud on AWS, Azure, and GCP, as well as on high-performance computing, or HPC, environments. Over the next few slides, I'll describe the process of setting up and running the pipeline on on-premises HPC. In order to set up and run the PacBio WGS variant pipeline, there are essentially 3 steps. Set up the environment, by which I mean setting up the basic tools you'll need to run any WDL workflow. This involves choosing and installing a WDL execution engine, choosing and installing a container runtime, and configuring the WDL engine to work with your container runtime and cluster job scheduler. Next, download code and resources, by which I mean downloading the specific code and reference data used for this workflow. And finally, setting up an individual analysis run, by which I mean creating a file, defining the inputs, and running the workflow. Workflow description language is an open standard for describing data processing workflows with multiple options for execution engines. On HPC environments, Cromwell or mini-WDL are typically used. Cromwell is a flexible and highly configurable engine written in Scala. Using a database backend, it supports call caching, or storing the results from a task to avoid unnecessarily rerunning the same task. It has 2 modes, a single workflow run mode and a persistent server mode, where jobs are submitted via a REST API. Mini-WDL is a Python implementation with a simple base configuration and a plugin system. Mini-WDL supports call caching with a file-based backend and has only 1 mode, a single workflow run mode. Cromwell and mini-WDL are both great options, and there really isn't a one-size-fits-all recommendation. For example, while it's often easier to get started with mini-WDL, Cromwell is probably a better choice for high-volume deployments. Dependencies for our workflow and many other workflows like it are provided in containers for improved reproducibility and easy deployment. We generate OCI-compatible Docker containers and deploy those to key.io. Key is spelled Q-U-A-Y. Cromwell and mini-WDL or any of the cloud WDL execution services will download, cache, and manage these containers and execute tools in them using runtime such as Docker or Aptainer. Aptainer, previously known as Singularity, is a container runtime used by many scientific computing environments. Aptainer and other runtimes like Podman can run OCI-compatible containers from Docker. The choice of container runtime has security implications and is typically best left to your HPC system administrators. While the PacBio WGS variant pipeline can be run on a single compute node with sufficient CPUs and memory, it contains many parallelizable steps and will perform best when scheduled across a cluster using a job scheduler. Cromwell and mini-WDL support most of the commonly used HPC job schedulers. Cromwell has a very flexible support system via backend configuration, while mini-whittle typically depends on additional plugins. Links on this slide point to the configuration options or plugins. Once we've set up a whittle execution engine, we need to download a copy of the workflow. The best way to do this is to clone the GitHub repository using a command like the one on this slide. I'm going to highlight a few of the bits of this command just to understand them better. To ensure reproducibility, our releases are tagged with a version number, for instance here, version 1.1.0. So we need to make sure to include the branch flag when we clone this repo to get this version of the workflow. We also use the Git submodule system to share some common tasks across workflows. So in order to make sure we clone these correctly, we need to make sure to use the -- recursive flag and to update any submodules if we're pulling updates to the workflow. The PacBio WGS variant pipeline has been built to work with the GRCH38 no alt analysis set reference. All reference data, including the reference sequence, index, annotations, and VCFs with allele frequencies, have been bundled and uploaded to GCP, Azure, AWS, as well as Zenodo. For on-premises use, we need to download this file and decompress it onto the file system where the analysis will be run. And we need to note that file path for use later. Next, we need some input for a run. So a cohort in our terminology consists of one or more related samples. The workflow will work for single samples, which we call singletons, duos, trios, quads, etc. Above 5 samples, you may need to tweak some of the memory for joint calling. For each sample, though, we need 2 types of input. Data, that is one or more unaligned HiFi BAM files totaling about 30-fold depth. The best practice here is to use standard Revio WGS output with 5MC base modifications, but without the consensus kinetic calls. We also need metadata about the sample and family. So that includes things like sample identifiers, sample sex, phenotypes encoded as HPO terms, and relationships to the proband. So that is whether the sample is the proband, the mother, or the father. If you don't have your own HiFi WGS data, you can get some example data for the genome in a bottle Ashkenazi trio from the websites at the bottom of this slide. I'll be using that in the examples that come up. Now that we have the reference data and some sample data, we need to prepare the inputs definition files, or inputs.json. You can grab a copy of this template from the repo you downloaded earlier at the path shown here on the screen. JSON files are a little picky, but you can find command line or web-based tools to verify that your file is a valid JSON file. These JSON files contain 3 basic types of information. The first are paths to the reference data that you're going to be using. So this is data that will not vary between your runs. The second part are information about where and how you want the pipeline to be run. And the third part is the variable information about the specific samples in the family you're going to be analyzing. This stuff is typically under the human WGS.cohort section. The first thing we're going to do is update the path for all the reference data. So this is the static information that will be the same between runs. And we're going to replace this placeholder, local path prefix that's in the template files, with the path that you're actually using. So in this case, I replace local path prefix with path to dataset. And I usually just do a search and replace across the entire template. Next, you're going to delete any of the rows with optional fields used for overriding defaults, like increasing the memory for certain tasks or changing the deep variant version. These all have defaults that are set in the workflow, so we're just going to wipe them out. Take out the row entirely. Once the changes have been made, save a copy of this file somewhere on your disk. If you're trying to understand the input types and the allowed values, these are detailed more in the documentation described or linked at the bottom of the page. Next, we'll fill out the cohort-specific values for our test run. In the example here, I'll be setting up a singleton run for the HG002 sample. First, make a copy of your modified template from the last step. In this copy, we're going to replace some of the values and delete some of the others. So we'll start by replacing the cohort ID and the sample ID. In this case, since it's a singleton and we're only analyzing one sample, we're just going to replace it with the same value in both cases, HG002. In general, for cohort ID and sample ID, try to use just alphanumeric characters, underscores, and dashes, as these are commonly used in file names, and we don't want to put anything that could interrupt the file name, cause problems later. These should be double quoted strings as well. Movie BAMs are a comma-separated array of double quoted strings. So what that means is you can have more than one input unaligned BAM for each sample. Each of these BAMs should have double quotes around them. And if there are more than one of them, we need to have a comma separating them. In this case, we have just one BAM, and it's got the local path on the disk to the BAM that will be used for this sample. Next is the sex. This is the reported sex, and it's either all capital letters male, all capital letters female, and double quoted. You'll note that anything here in the input template with a question mark is an optional value. So we could not provide the sex as well. Sex is used for a couple of the samples for genotyping. And if sex is omitted, then it's assumed to be an XX karyotype. Next is affected. Affected is a Boolean. So that means it's either going to be true or false without quotes, all lowercase. And at least one sample in a cohort must be affected if you request tertiary analysis downstream. Next, mother ID and father ID. Again, you can see the question mark indicates that these are optional fields. And since this is a singleton input and I'm not providing data for the mother or father, I'm just going to delete those completely. If they are included, these should be the sample IDs for the mother and father that are included in the same run. Finally, phenotypes is a comma-delimited array of double-quoted HPO terms. HPO is a formal ontology of human phenotypes, with phenotypes represented by HP and 7 digits. Those terms can be obtained from hpo.jax.org, and HPO terms are used for tertiary analysis, but you can always use the base term HP 6 zeros and a 1 if you don't have anything more specific. In the example on the right, I've changed the sample and cohort IDs to HG002, changed the sex to male, changed affected to true, provided a UBAM, an unaligned BAM as the input, and added the base phenotype HP000001. Finally, you're ready to launch a job. The exact syntax will vary, but typically the command will require paths for 3 things. So it's going to require a path to the configuration. This is the configuration file we set up to run it on this cluster. It's going to require a path to the inputs for this run, and it's going to require a path to what's called the entry point into the workflow. So the entry point into the human WGS workflow is this main.whittle file. It'll look a little different for Cromwell, and this inputs line is going to vary on every run with your new input data. On a high-performance computing cluster with sufficient resources such that there are no bottlenecks, analysis time for a singleton is around 17 hours, and for a multi-sample cohort, around 22 hours. Many output files are generated by the workflow, and I'm not going to take the time to go through all of them, but at a quick summary level, at the sample level, there are alignments, variant calls, and statistics. At the cohort level, if you have a multi-sample cohort, you'll have joint called small variants and SVs, and some additional statistics. And if the optional tertiary analysis has been enabled, the outputs will include filtered annotated rare variant calls for small variants and SVs. Alignments, variant calls, and genomic interval files like bed files can be viewed in IGV, and structured or tabular data can be viewed in Excel or text editors. Some tools, like Target, have their own special tools for visualization, like TRVZ. Refer to those repositories for those tools to learn more. A detailed description of all file types that are output by the workflow is available in the pipeline documentation, linked at the bottom of the slide. It can be useful to have an idea for how many variants are expected to be called for each sample. While this can vary widely depending on ancestry, a survey of over 150 diverse samples from public consortia yields an average of 4.8 million single nucleotide variants, 1.1 million small indels, 25,000 structural variants greater than or equal to 50 base pairs, around 50 CNVs of greater than 100 kb, and phase block NG50s of approximately 1.2 megabases. The tandem repeat genotyper tool target works off of a list of input loci, and you can choose from existing input files covering 56 loci with pathogenic expansions, 170,000 polymorphic repeats, or 900,000 polymorphic repeats. So the output here is going to depend on what input file you use. Our genotyper for genes in segmental duplications, paraphase, currently types 9 genes in segmental duplications, but will be updated soon to cover over 160 segmental duplication regions. Finally, if the input BAM had 5MC base modification sites generated by the tool Jasmine, as is typical for all whole genome sequencing data generated on Revio, you'll have methylation information for over 28 million CpG sites. We have a lot planned for this workflow in the next year. In addition to bug fixes and maintenance updates, like improving platform compatibility and updating to newer versions of some of our tools, we'll also be adding new types of analysis. We plan to add pharmacogenomics typing, PGX typing, with tools like Pangu, pb-Starphase, and PharmCat. We have plans for calling homoplasmic and heteroplasmic variants in mitochondrial DNA. We have some team members working on our next generation of structural variant discovery tools. And finally, we'll be adding tools for the analysis of atypical or differential methylation. So keep an eye on the GitHub link at the bottom of the page for new releases, and check in with us if you have any suggestions or questions. Thank you for your time and interest in the PacBio WGS variant pipeline and associated tools.
Thank you, Billy and Yanan. I think we have time for a couple questions. Just as a reminder, if you do have any questions, please just put them into the question box on your control panel. Yanan, I think first I have a question regarding deep variant and Google Cloud, maybe specifically about the cost. Can you maybe outline about how actually deep variant, about running that, say, locally versus on an HPC versus cloud and licensing costs?
Yeah, absolutely. So the pipeline itself, including all the components, is designed to be infrastructure agnostic. That means you can take the pipeline and its associated tools to run on HPC on-prem or choose any of your favorite cloud. If you're running things on HPC and things are already established, there's actually no additional cost. There's no paid license fees we required for the pipeline itself. If you choose to run in any of your favorite cloud, GCP or AWS or Azure, there will be associated billings about compute and storage.
Thank you. Billy, there's a question. Do we implement a sex check in the pipeline?
So at the moment, the sex is only used for genotyping for HiFi CNV and for target to adjust the genotype for variants in the allosomes. There are no checks to compare the inferred sex to the reported sex at the moment. We had some of those previously in the SnakeMake version of the workflow, and we may add those back, but right now it's just used to get the correct genotype.
Thank you. Also a question. So is this pipeline available to work on other genomes, whether that is other human builds or other species?
Right, so that's a really good question. So a lot of the tools in the pipeline have been primarily, have been designed to work originally with GRCH38. So some of these tools can be applied to other references as well. Some of them we already have some of the resources that are required for that, the resources being like bed files and things like that. Others, those resources haven't been developed yet. So you could, for instance, Target has resources for GRCH38 at the moment. Paraphase has resources for GRCH38 and some for HG19 as well. So individual tools may work with different references. The workflow itself is a human workflow, and it's entirely built to work with GRCH38. And that's just because we're going for something compatible with all the tools in the workflow. As more resources for these other references become available, we may be able to expand it to work with these other references. But for the moment, it's only GRCH38.
Great, thank you. Yanan, I think it was covered on one of your slides, but maybe you can expand or just recapture the material. So in terms of high phase versus WhatsApp, what does high phase do that WhatsApp does not do?
Yeah, it's actually a pretty good question. So WhatsApp has been a pretty popular tool so far, what we have seen with the solid performance and a tool from the community. But with HiFi reads, there are so much potentials we can still to explore and understand with more complex questions, especially structural variants. That's we see there is additional need for phasing, and that's how we develop HiFi in-house. So HiFi will be able to jointly phasing not just small variant as well as structural variants. And we're also seeing a performance improvement, including like phasing blocks and overall accuracy as well.
Great, thank you. Yanan, can you also comment, do we have recommendations for more advanced tertiary analysis?
Yeah, absolutely. So we welcomed two tertiary analysis partners last year, so Genix and Golden Helix. So if a user is interested in better understand how the biological knowledge and discoveries within the variants, we would recommend to go with one of the two partners. They provide excellent services, especially in rare and inherited disease for whole genome.
Billy, there's a question about recommended coverages. Can you comment on recommended human genome coverages and anything about going less than 30x?
Yeah, so that's a good question. We typically recommend and target 30x coverage, but we regularly use this for as low as 20x coverage or something like that. This pipeline was primarily designed as a research tool for rare disease research, and you really need that depth to make sure you're not missing variants, to make sure you have nice recall, especially if you're looking at trios and you're running the parents as well. You want to make sure you're not calling something de novo that exists in the parents. We're thinking about extending it to work with lower coverage. We may also just fork it into a slightly different tool at some point. These are ideas for down the road, but we may make it work with lower coverage later. At the moment, though, we target 30. We use it as low as 20x.
Thanks. I know you covered it. Maybe you can recap maybe a bit about Mini-Whittle versus Cromwell and the different job schedulers.
Yeah, so I think first though there's a slide earlier in the deck, and so hopefully you'll get a copy of the deck and the video in the future, and you'll be able to look at this slide and find some links to some useful tools. I think the question here specifically was about the PBS scheduler, and I don't think I had it on that slide. With Mini-Whittle, working with different job schedulers is handled by different Mini-Whittle plugins. The plugins are not terribly complicated to write if you write Python and you can look in an existing plugin. But if you're not comfortable writing a Python plugin, I think for PBS I would go with Cromwell. So Cromwell is sort of eminently, it's very configurable. You can put anything you want in the little command callers, and so you can take your PBS commands and just drop them right in the quotes in the thing that gets sent to the job scheduler, right? So for a tool that doesn't have a Mini-Whittle plugin already available, I think Cromwell is probably the way to go, and it's going to take a little bit more configuration, at the start, but your use case will definitely be covered.
Wonderful. Billy, can you also comment, are customers already using this pipeline?
Yep. We have several customers that have transitioned from using the SnakeMate version of the workflow that we've had out for a while now to this, and then to this Whittle version in the past year. I think there's at least 10 in the US, at least 10 in the Asia Pacific region that I know of, and many in the EMEA region as well. So we have a lot of users that have transitioned over.
Great, thank you. I think with that, we'll start wrapping up here. I'd like to thank our two speakers, Billy and Yanan, for sharing their expertise and experience today, and I'd like to thank you all for joining us. In closing, wanted to briefly mention 3 things. First, in the next week or so, we'll send you an email with a link to today's recording, should you want to go back and listen to any part of it again. Second, please keep up to date on PacBio webinars and events by visiting and bookmarking pacb.com/events. And finally, immediately following this webinar, you'll receive a short questionnaire. Please do take the 2 minutes that is required to fill it out, as it helps us understand your needs and also plan for future webinars. So thanks again for joining us today. I hope you've enjoyed this, and we hope to see you in future webinars. Take care. Bye.